Hello children, welcome to NEET AIPMT Foundation Biology online classes. This is plant breeding and crop improvement part 4. Children, we are discussing about the crop improvement methods, isn't it? So, today's class we are going to have one more method of crop improvement. So, before going to that, let us briefly discuss some key facts. The plant breeding can be done for disease resistance. Do you agree? Yes, we have to. A wide range of fungal, bacterial and viral pathogens affect the yield of cultivated crop species, especially in tropical countries, tropical climates. So, in this situation, breeding and development of resistant to diseases enhance the food production. Is it not, children? Next, plant breeding can be done for insect pest resistance. You know very well, insects and pest infections are two major causes for large destruction of crop plants. Insect resistance in a host crop plants may be due to morphological, biochemical or physiological characteristics. Okay. So, plants with resistance are developed by plant breeding methods. Okay. Next, plant breeding can be done for improved food quality. Improved food quality. So, breeding crops with high levels of vitamins and minerals or higher protein and healthier fats is called biofortification. The plant breeding is undertaken for improved nutritional quality of the plants with the objectives of improvement in protein content and quality, oil content and quality, vitamin content and micronutrient and mineral content. Okay. Single cell protein, that is called SCP. So, one of the alternate sources of proteins for animal and human nutrition is single cell protein. Okay. Spirulina, a cyanobacteria like spirulina can be grown on the wastewater from potato processing plants containing starch, straw, molasses, animal manure and even sewage to produce large quantities of food rich in proteins, minerals, fats, carbohydrates and vitamins. Okay. So, algae, bacteria, fungi used as a food or feed. So, the tissue culture is another. Plant tissue culture is a technique of maintaining and growing plant cells, tissues or organs aseptically on artificial medium in suitable containers under controlled environmental conditions, that is called in vitro. Okay. So, tissue culture is a wonderful technique for the production of large number of plants in the short span of time. Okay. Right, let us proceed, children. Are you ready? Let us proceed. Polyploidy breeding, the technique polyploidy breeding. The term ploidy represents the number of chromosomes present in that particular organism. Okay. So, polyploidy breeding, we are going to discuss about polyploidy breeding, an organism in which number of complete chromosome sets is more than the diploid number is called polyploidy. Okay, what is diploid? Diploid means 2n. The more than diploid number of chromosomes, set of chromosomes is called as a polyploid condition. It is a useful practice to express the chromosome number of an organism in diploid, which is symbolically represented as 2n. You know, children, the more the number of chromosomes, the more the number of characters. Okay? So, in all sexually propagated crops, such expression is of specific significance, because the pairs of chromosomes indicate the number of chromosomes in each genome. Okay. The next slide we are going to see what is the genome. Okay. The organisms are able to maintain chromosome numbers over generations due to the fact that the gametes have n number of chromosomes, while all other cells of organisms have 2n number of chromosomes. Okay. So, here some representation of the chromosome in a particular organism. So, a condition 2n minus 1, it is called monosomic condition. Okay. 2n condition is diploidy. 2n minus 1 monosomic or monoploid. 2n plus 1, this condition is represented as trisomic. Trisomic. So, let us know the difference between a genome and a gamete. Okay. What is genome? What is gamete? What is the difference between the genome and gamete? In sexually propagated crop, a gamete consists of n number of chromosomes, means the number of chromosomes represented with the letter n, representing all genetic characters of such crop. Such a crop chromosomes carrying specific genes is called a genome. Okay, the group of chromosomes carrying specific genes is called a genome. Then a gamete is a reproductive structure carrying genome. The gametes from male and female fuse to form zygote. Gametes are found in the pollen and in ovule. Pollen means male gamete. Ovule, the female gamete egg is present in that ovule. Okay, in the pollinated crops, ova, sperm. Ova means egg, the sperm in the mammalians. Okay, in animals, gametes. Okay, gamete singular, gametes plural. Okay. So, however, some organisms may have more than 2n chromosomes. Okay, if 2n is present, diploid condition. More than 2 it is present, then it is called polyploid condition. The polyploids may be triploids, 3n condition, tetraploids, 4n condition, hexaploids, 6n condition. When polyploidy occurs within a species, it is called as a autopolyploidy within the same species. When a polyploidy occurs due to the chromosomes from two species, then it is called allopolyploidy. Autopolyploidy within the species. When it occurs due to the chromosomes from two species, then it is called allopolyploidy. The polyploidy may be caused by fertilization of an egg by more than one sperm, failure of meiosis during the gamete formation. So, in normal conditions this polyploidy may not occur, it is an abnormal condition we can say. Okay. So, this to obtain to do this the protoplast culture, protoplast technology has opened up avenues for the development of hybrids of even asexually reproducing plants also. The somatic hybrids may be used for the production of useful allopolyploids, Okay, there are various applications are there. This protoplast culture is one of the technique in tissue culture. There are several advantages are there. Okay, we will discuss it later. The polyploidy can artificially be introduced by a chemical called colchicine. Colchicine is the chemical which causes mutations, which causes the change in the structure of genes, structure of DNA. Okay, so by adding this chemical colchicine, artificially polyploidy may be developed, or by crossing diploids with tetraploids. Okay, this is also another technique. So although the triploids exhibit sterility, the polyploidy is useful in some ways due to some advantages. Okay, so polyploidy examples let us discuss. In some plants like apple, pear, the fruit size is bigger in triploids, means as the number of chromosomes increased, so the size representing chromosome developed in these polyploids. Okay, so triploid condition, normal condition is diploid, here the one chromosome is added. Triploids, the size of the fruit is bigger in triploids, with in which apple and pear. In sugar beet, the high sugar content is associated with triploids, triploidy. So here sugar content is increased. So ploidy breeding involves hybridization and the selection of plants, varieties, species exhibiting various polyploidy. Okay, a great deal of success in the wheat breeding was achieved when origin of hexaploid the wheat was discovered. Hexaploid wheat was discovered. Hexaploid wheat was a result of sequence of natural crossing and chromosome doubling of chromosomes. Okay. In the study of crop evolution, many such instances are recorded in crops like potato, paddy and sugarcane. So it is breeding most commonly used when the desired variation is required to be induced in a vegetatively propagated crop. Okay, it is the breeding most commonly used. Which breeding? Polyploidy breeding. Okay, in which when the desired variation, means whatever you want, the good quality is required, it can be induced, okay, in a vegetatively propagated crop. Vegetatively propagated crops means other than flower, if the plant propagate with the other parts other than flower, such plants, such a group of plants come under the vegetatively propagated crop plants. Okay. The next one is mutation breeding. What is mutation breeding? What is mutation? First of all, you have to understand. Mutation means it is a sudden heritable change in an organism is called mutation. Okay. So induced mutations, the mutations which are artificially created, it is possible to induce mutations artificially through use of chemicals, okay, like colchicine, or by use of radiations like gamma radiations, and selecting and using the plants that have the desired characters as a source in breeding. This process is called as mutation breeding. Okay. For example, in mung bean, resistance to yellow mosaic virus and powdery mildew, these are the diseases, yellow mosaic virus disease and powdery mildew disease, okay, were induced by mutations. Okay. Sharbati sonora and Pusa lerma are the two important varieties of wheat produced by exposing Sonora 64 and Lerma Rosa 64 to gamma radiations. Okay, it is Mexican dwarf wheat varieties they are. Okay. So let us know about this mutation breeding. Mutations are defined as heritable changes in the structure, number of genes or chromosomes. Heritable means which can be passed from the parents to the offsprings. So, the mutations can be natural, they are called spontaneous mutations. They may be artificial, such mutations are called induced mutations. Many natural mutations are recorded, but the stability of mutant means the substance or the particle or anything which cause mutations are called mutants. Okay, the stability of mutant expression needs a thorough testing over generations, as many mutants have tendency to revert to normal, because there is a chance of reverting again, come back to the normal. So, that is why the stability of mutant can be tested thoroughly over generations. The mutation theory was proposed by Hugo de Vries. Okay, Hugo de Vries proposed the mutation theory. The mutations can be induced by what are known as mutagens. Mutagens are the agents which cause mutations. The several mutagens like X-rays, UV rays, gamma rays, and some chemical substances have been extensively used for inducing mutations. Okay. The success in using mutation as a breeding method has been reported in some crops like barley, wheat, rice, groundnut. Okay. The wheat, just now we discussed the Sharbati Sonora and Pusa Larma wheat varieties, okay, dwarf. So, at the molecular level, point mutations are due to the alterations of sequence of nitrogen bases in the DNA. Okay, that is guanine, adenine, thymine, cytosine. There are some nitrogen bases with which the DNA, okay, is made up of. DNA is nothing but the genes. Okay, the mutations which occur in the chromosomes, they are called chromosomal mutations. The mutations which takes place in the genes, the gene mutations. Okay. So, the importance of plant breeding, it is very interesting and very important to know about the importance of this plant breeding. So, it produces more productive crop plants, and thereby improves the food production. So, it can produce crop plants with superior features like fruit or grain size, vitamin content, etc. It can generate disease resistant crop plants, as for example, the rice plant against the blight disease, tomato against the wilt disease, sugarcane against the red rot, okay, which are caused by these pathogens, bacteria, fungi, virus. Okay. So, the plants with resistant genes or disease resistant variety plants can be produced by using this plant breeding techniques. Okay. The beginning only we have discussed. And another advantage, the plant breeding may also lead to tolerance against the extreme dryness or wetness, means drought resistant varieties can be produced. It helps in stabilization of characters in a particular crop plant. It results in the production of new varieties of crop plants, and it results in the expression of heterosis or hybrid vigor. Okay. It involves the adaptation or acclimatization of a plant species to changing environmental conditions. Okay. When a hybrid is produced by the fusion of somatic cells of two varieties or species, it is known as somatic hybrids. The somatic hybrids are produced by a technique called somatic hybridization. So, the advantage, the use of plant cells to generate useful products, a plantlet which is used for various purposes using the tissue culture technology. Okay, the plantlets are used for rapid clonal propagation, okay, clonal selection. Then transgenes can be introduced into the individual plant cells. The plantlets can be regenerated from these cells which give rise to highly valuable transgenic plants. Okay. For induction of mutations, just now we have studied the mutation breeding. For the induction of mutations, as genetic manipulations can be carried out more rapidly when the plant cells are in a protoplast state. New genes can be introduced, male sterility, herbicide resistance. For raising stress resistant plants also, these plant breeding techniques can be used. Okay children, so keep reading the book. Thank you for watching our video. Have a nice day.